I'm in Madrid for the 12th EACPT Congress with three of our major award winners.
Yes, I'm Nick Bateman. I'm from Edinburgh and I'm Professor of Clinical Pharmacology.
I'm Christoph Schneider from the University of Bern in Switzerland, and I'm a PhD student.
I'm Antoine from the University of Liverpool. I'm also Centre for Drug Safety Science and I'm a lecturer in pharmacology.
Nick, would you start by giving some idea of the importance of paracetamol poisoning?
Paracetamol is the commonest poison we see, and it's the most frequent cause of acute liver injury and overdose. And it's a problem right across Europe. Even though the incidence of different countries varies, it affects all countries in the European community.
And what was the key issue you were exploring that led to the paper in the Lancet you published?
The major problem was that we've got a treatment for paracetamol poisoning using intravenous acetylcysteine, and this causes major problems with adverse events in a proportion of patients. And the current regimen is also 21 hours, and we think we could shorten that. This causes patients not to like the therapy and to get adverse events, particularly vomiting and anaphylactoid reactions. And so we reckoned we could shorten the regimen to 12 hours from 21 and also try and reduce the frequency of adverse events by a shortened regimen. And that's what we managed to do.
So what were the key conclusions of your paper?
Well, the key conclusions were that we could reduce adverse events by 3/4 for anaphylactoid reactions and about 50% for vomiting by using a shorter and simpler regimen. And we think we'll be able to discharge patients much earlier because we've now got new biomarkers which should allow us to identify patients who are safe to be discharged much earlier in their course of therapy.
And what stage are you be able to influence health policy with your work?
We are currently engaging with the MHRA and others and beginning to introduce this protocol in clinical practice, and we're beginning to do it in Edinburgh and want to spread it around the UK in a formal study to try and work out where the treatment is safest and where there might be any particular problems. It's possible that patients who present later, for example, might be more appropriate on a more intensive acetylcysteine regimen, but this requires further work.
Nick Bateman, thank you very much indeed. Christoph, would you explain the background to the work which led to your Translational Award?
So we were interested in the interaction between carbohydrates and proteins, and the problem was for a long time that it was not really possible to assess interactions of carbohydrates because you were simply not able to synthesize them. And now the technology of the Consortium of Functional Glycomics provides a tool where you can assess 610 different glycans in a ELISA type assay, and we were the first ones to assess the IgG mediated binding in such a big screening.
And what's the clinical relevance of the work you've been doing? Which sort of diseases are relevant to this work?
So glycans are basically everywhere. Every cell is surrounded by glycocalyx, and therefore also many diseases are affected by changes and by interactions of glycans. For example, tumor cells have differently expressed glycans on their surface, and for our study it was also important that many bacteria and viruses use carbohydrates in order to attach to a host cell, so the very initial step of infection is mediated by glycans often.
Was this a finding where you find an interaction with all types of immunoglobulin, or was it a subset which is relevant to what you were finding?
Well, mostly it is mediated by IgG2, but we have attachment sites which are also recognized by non-IgG2 isotypes, so we cannot exclude or really say it's only IgG2, it's for sure IgG, and therefore it's also included in IVIG since it contains all the 4 different IgG isotypes.
And finally, what do you see as the relevance to clinical treatment in future of your work?
I guess we have like 2 relevant discoveries we made. So first of all, we could link the immunogenicity of a glycan to certain structural features, and we think that in terms of glycovaccination, this could be of use to, in order to design structures which then also have the potential to raise an immune response. And on the other hand, we saw this response against attachment sites, where we believe that you could use polyclonal antibody preparations in order to block the adhesion of microbial structures.
Thank you very much indeed.
Thank you.
Daniel, would you describe the context for the work which led to the publication which gave you this translational award?
Yeah, so drug-induced liver injury is a huge problem clinically, but also a big problem for attrition within drug development. One of the major problems that we see clinically is paracetamol overdose.
Why do we need new biomarkers of liver injury?
So currently the problem with paracetamol overdose is that the majority of patients present to hospital very early on before the current tests, alanine aminotransferase and synthetic liver function markers, are elevated in blood. So we can't confidently diagnose liver injury or exclude the development of liver injury. So there's a real need for the development of new biomarkers that are with enhanced hepatic specificity and sensitivity to replace the current tests.
What kind of pathologies were you exploring which might give you confidence these biomarkers might be of interest?
So the mechanism associated with paracetamol-induced hepatotoxicity is linked to apoptosis, necrosis, mitochondrial dysfunction, but also the activation of the immune response. And we have a panel of biomarkers that reflect those different mechanisms, such as mitochondrial dysfunction, reflected by glutamate dehydrogenase. Apoptosis and necrosis are determined through keratin 18, and HMGB1 is a reflection of the activation of the immune response. But also the biomarker microRNA 122 has enhanced hepatic specificity compared to the current tests.
And what were the key messages in the paper?
The key outcomes of the paper and the conclusions are that this panel of biomarkers were elevated in the blood of patients that subsequently went on to develop acute liver injury, and they currently were able to detect the presence of acute liver injury before the current tests.
Do you think this is likely to be paracetamol specific, or are you already exploring other sorts of liver insults where this might be a useful clinical advance?
So we've looked at a number of these biomarkers in naturally occurring liver diseases and liver disease associated with alcohol, but currently we're working with a number of pharmaceutical companies as part of huge IMI-funded consortia such as SAFETY, and also American-based consortia funded through the Critical Path Institute to try and enable the future qualification of these biomarkers by regulatory authorities.
Daniel, thank you very much indeed.